Thanks a lot to the organizers for allowing us to present in this event our work on genomic prediction of climate change adaptation in beans. The motivation behind is to infer local adaptation in wild beans by looking at the environments where those beans come from. And this idea has been built since Humboldt's writings, in which he reflects that geography environments exerts its influence on plants. Therefore, nowadays we should be able to go back and infer how nature influenced the adaptation in those regions for the local ecotypes we find there. Under this idea is that we approach the current dataset, which involves wild beans across the America, the natural distribution range for the species. Wild beans are very diverse in terms of size and color in their seed coat, but they are also very diverse in terms of the environments they occupy. So when we look at the wild beans, which are blue dots in this map, and we try to look them across the Americas by looking at the precipitation in those regions, the annual precipitation, we might start seeing that Mexican wild beans tend to be in places where there is more apparently dry environments compared to beans that come from South America, such as Colombia, Ecuador, or not Peru. This is just by looking at the precipitation, but of course, draw stress is perceived as a balance in which water not only comes into the system by precipitation, but it's also loosed from the system by evaporation and transpiration. So that's why we do not only look at the annual precipitation patterns, which are summarized here, but also try to look at a couple of indices that combine those two properties, how much water the system receives, how much water the system loses through evaporation and transpiration. And by looking at those draw index, we might find again that beans from Mexico tend to be apparently more draw adapted than beans from Colombia and South America. So this data set, which is, as you can see, built not on raw bioclimatic variables, but instead on ecophysiological models, is what enables us to build a predictive model on the local adaptations in those sites. And again, I'm insisting we are working on wild beans because for those ones, it's possible to assume that competitive testing has been testing those plants more than persistence. Persistence is the case of the cultivated beans that are probably in these regions because farmers brought them very recently, as compared to wild beans that have been under competitive testing, in this case for drought adaptation during many generations. So based on that assumption is that we perform this first genome environment association, looking first at drought tolerance, computed by the indices I just showed you. And interestingly, we see here marked in red the different regions and SNP markers that associate with the draw index inferred with environmental variables. And if we move across chromosome until the 11th chromosome, you might realize that this system is highly polygenic, meaning there are many regions with low to moderate effects influence the adaptation to drought tolerance in wild beans. This brings us different questions, such as how this system arose in first place. Was it perhaps a mutation that later on served as a platform for other mutations to build upon? And also, how can we predict adaptation when the system is so polygenic, such as this one? So we will explore these questions, but before I would like you to reflect on the nature of this variation and what's perhaps the driver behind this adaptation. And by that, we compare different summary statistics between associated and non-associated regions. Associated regions tend to be more in highly SNP density windows, also with slightly high nucleotide diversity, lower theta Watterson's statistic, and the combination of these two leads that associated regions are generally under higher Tajima's D scores, which can be interpreted taking into account the mapping exercise and this exercise use population structure as covariables to divergence selection towards drought tolerance. Also look at this. This can be interpreted as a background selection across the genome, any type of signature associated, for instance, with adaptation, mainly derived from the gene pool structure that is observed in the species. In other words, this conclusion leads that drought tolerance might be beneficial under drought stress, but as expected, is not necessarily so good under well-irrigated environments. And this is the typical trade-off we would expect for a locally adapted trait. Genome-environment associations, as I said, are controlling for potential spurious factors such as demography and the background selection in the non-associated regions. But what happened when we look not just at drought tolerance, which concluding seems very polygenic and being under divergence selection, but instead we look, for instance, to heat tolerance in the same panel. In this case, the index was built more in a more simple way. It was simply in the cases in which temperature in the evening was higher above a physiological threshold for the species. And that is because if the threshold goes, the temperature in the evening goes above the threshold, the pollen loses viability. So we map again those regions across the 12 chromosomes, using different algorithms to inform the genome-environment associations here in different colors. And again, we find ourselves with a highly polygenic landscape, which does not necessarily correlate, as we have explicitly carried out, with the previous one. So it's not a case of a polygenic pleiotropic adaptation. It's completely different adaptive data sets that are being involved in this combined response. Here in red asterisk, we see genes that have been previously described as heat shock proteins and that correlate with these associated regions. So this also serves as an ad hoc validation for this analysis. So we have these two scenarios in which we have adaptation understood as drought tolerance, as heat tolerance across wild beans in the Americas. We try to understand a bit how it was built, and it's related with this divergent adaptation across environment, which implies a trade-off for adaptation and yield, which is what perhaps sometimes is more interesting for future applications. But still, we have a remaining question, and is how can we predict adaptation in the system? And here is what we are currently doing, and that kind of pushes the field forward and is trying to estimate genomic estimate adaptive values as a proxy to what before was known as a regions of known adaptation, of prediction of known adaptation. Instead, genomic estimate adaptive values can be understood more related to what in plant breeding is called genomic estimate rating values, or in medicine is called polygenic risk score. So all those three are estimates that assume a polygenic landscape versus the risk of no adaptiveness, which assumes a more Mendelian determination of the adaptation. So here we move forward into a validation set, and we build interspecific hybrids between beans, common bean and Tepary bean, being Tepary bean known as our source of heat and drought tolerance. Those crosses were back-cross complementary and tested in 4 localities for 4 yield traits and across more than 15,000 SNP markers. Again, the combined landscape is highly polygenic across different localities, which are marked here in color and symbols. That meaning that not only we have a polygenic system, but it's a system that reflects a lot of genome environment interaction across the regions. So building the prediction here is much more difficult if we rely on traditional methods, and that's why we move into these genomic estimate adaptive values. Unsurprisingly, our predictive ability, that is how much correlation we found in our testing data set comparing the prediction and the observed real value, we are reaching for different stations, the multilinear stations, up to 75%. This is only for one of the four traits that was examined, each of these lines, but across different lines and colors, we have the different traits. Again, for another locality, nearly 75%. And if we zoom in into these regions, we reach the saturation very early on. With only 250 SNP markers, we are already reaching the saturation of the prediction. That meaning that we know adaptation in wild beans is very complex, involving a lot of regions, polygenic regions, and genotype environment interaction. But still, we are able to successfully predict the adaptations in those regions by using genomic prediction. In this case, a Bayes algorithm. Difference is that we are not estimating a breeding value, but instead an adaptive value. This led me to close with another citation. This book, Darwinian Agriculture, Natural Selection has already tested more combinations than humans ever will. And therefore, if we have a polygenic system such as this one, we might learn more by looking at the system and comparing it across regions and species rather than trying to replicate it. So as conclusions, genome-environment associations and genomic estimate adaptive value enable exploring and predicting the genetic basis of adaptation in wild beans. Both approaches utilizes mixed linear models as response variable. They are including not a phenotypic variable, but instead we suggest an environmental index as compared to a raw climatic variable. It is advisable, we insist, to use these physiological-based indices since they are more informative of the processes we are looking as part of the adaptive response. Combine that with summary statistics to interpret the cause behind those selection signatures and try to predict those responses using genomic prediction values seem to move forward the field from interpreting and staying on genome-environment associations and trying to leverage those materials as predictive scores. We have applied this, both genome-environment associations and genomic estimate adaptive value in common and Tepary beans to describe the architecture to both draw and heat tolerance, but more importantly, to predict the potential of abiotic stress tolerance in the species. Thanks a lot.